Welcome to our workshop. This is the third workshop that I've done on the topic, and it's an evolution of previous ones. Has anyone been here for our bioteam workshops years past? I'm not that good with faces. Oh, that's nice. Thank you. Too. That's great. I actually remember last year, and I was looking for him again this year, so I'm on the registration, the gentleman from CureVac. From Germany came, and I had previously, when I lived in Germany, applied to a job there. And I said, "Oh, well, now I'm giving you a workshop, so maybe you would hire me." He goes, "Well, let's wait until after the workshop and see." Anyway, so I wanted to see if he had come back to the workshop, but I guess he's not here. Anyway. Oh, there you are. You are my favorite bio-IT stories, but you didn't raise your hand, so. Oh, okay. It was a German hand raise. It was a very moderate German hand raise. Yeah, that's fine. Yeah, that makes sense. Okay, wonderful. So anyway, all to say is that it's nice to interact and it's nice to actually talk to each other. And we did make this workshop sort of build on our previous ones. The previous ones covered a little bit more sort of organizational things because we felt like our clients or the people that we talked to were really struggling with the beginning parts of bringing AI/ML into their organization. So even thinking about how to form teams around the concept, who was going to do the work, where they were going to fit it into their workflows, what kind of questions were appropriate to ask. So we had very much more so organizationally structured workshops, meaning like higher level strategy things, and all of that content is still on the Bioteam website. If anybody is interested, you can always go back or you can talk to us about those kinds of things later. This year, of course, things are changing so quickly, but even before this sort of explosion of tools that AWS is presenting, which we can talk a little bit about, Jordan and I really had the intention, let's see if we can, we'll do introductions in a second. We really had the intention to help folks come away from this with something tangible that they could actually feel like they made some progress in. We are not going to generate any chatbots today. We are not going to sort of really, promise you anything that's not deliverable, but we did want to make sure that we can get folks some hands-on experience, especially since open source models really are becoming much more available to your teams. And I know that there's always sort of a range of folks here from students to people actually working as data scientists who are probably much more capable than we are, to the leadership folks. And so I think it's important for us to level set at this point in time what it really means to go from having a model that is available that maybe your supervisor or you as a supervisor or leader say, now let's use it in our teams or in production or in our sort of research, to actually making that happen. And I think that that's a really important sort of jump that we as a scientific community have to make if we want to actually take advantage of these advancements, and maybe not the most recent ones, but also I think the most recent ones. I don't know, Jordan, if you want to add to that as a concept of like what we wanted to do here.
I think and just in general, trying to be a little pragmatic in how we adapt AI/ML technology for use in biomedical sciences, and also how to really make that compatible with the fact that data can be very messy in this domain.
Right. So that's sort of the goal of our few hours here together. And let's do some introductions. My name is Anna. I'm a senior consultant for Bioteam. I've been here about going on 3 years. So this is, as I said, my third workshop. And I started off in neuroscience. So I'm actually a scientist by training. I was in Tübingen in Germany doing my PhD. And then I realized essentially that nothing that I did in my work or in my postdoc, which was in immunology, was going to be relevant unless I figured out how to get out of Excel sheets and actually process data in a big way. And I teamed up with a startup that was working there. I don't know if anyone's in crop science here. Sometimes folks, I guess it's a life science symposium, but well, sometimes people come from crop sciences. So there was a crop science company that was actually doing machine learning predictions there called Computomics. And I paired up with them, and they were a great team, and they got me introduced to essentially getting teams ready to process large data through machine learning algorithms. And so I am not actually a data scientist or a bioinformatician, but I am a scientist who got into the field and became sort of familiar enough to make sure that the teams that I worked with were able to work with the analysts, and the analysts, good morning. And the analysts are more like Jordan, who's in our team. And that's sort of the fun part of being a bio team, is that we combine folks who really have that domain knowledge and folks who have the expertise in the technical field. And we sort of have them work together to explore concepts like machine learning or whatever is coming next. I think we'll be dealing with machine learning for a while, although the AWS presentation, this was funny. I won't say the company who had a slide that they were presenting for their colleague, actually. And they said it was sort of like a 5-year roadmap. And they said that in the next 5 years, they're going to, what was it? They were going to tackle LLMs. Something else about like a sort of deploy AI/ML and also get into quantum computing. And the guy who was standing up there was like, oh, yeah, no, this is already done. He was like, LLMs, we did this year. He's like, quantum computing, probably next year. And so that's, that's sort of the way that teams talk when, when it's not real, I guess. And so what we wanted to do was actually sort of bring us back to reality a little bit here. So that's introduction to me. Jordan, why don't you go introduce yourself a little bit?
Sure, I'll keep mine a little bit shorter. Nothing against you. I don't have much to say. So I started
data scientist, am I right?
Yeah, I know. I'm not very talkative at times. So I got my start in genomics. I realized very early on I'm terrible at wet lab work. So I transitioned over to using a computer, found out I loved it. So I really focused on bioinformatics. I then, for my master's, went on to do computer science and studied information retrieval. And I really wanted to take it back, a lot of the stuff that I learned, back to biology, back to bioinformatics, because I thought there was a lot we could still do. So thankfully, when I joined BioTeam, I didn't want to say the other thing, BioIT World, we're here already. When I joined BioTeam, I was really given an opportunity to work on things like visualizations and explore data. So I'm really happy to be here now, because I'm really excited to show you the stuff that I've done. So.
Can we get a little information also sort of about the crowd? I know we saw the registration list, but it's always sort of a toss-up of who shows up. Are we from the science side? Raise your hand, maybe if you're sort of more the science side, maybe more IT, bioinformatics, data science side. Awesome. Who's actually doing versus who's telling people to do it? So if you actually do it, raise your hand. Oh, fantastic. And are you telling people to do it? Okay. Good. So there's a mix. So essentially, I guess, probably the folks who are actually doing it, feel free to pipe up and say things, because I think it's really important that we level set as a community. I think some of the hype really gets people all frenzied. And I think especially scientists, when we do these sort of larger organizational assessments, scientists sometimes get really distracted from science when they feel like there's this pressure to do this thing that feels very nebulous and amorphous. And I think it's important that we sort of communicate to each other about refocusing on our expertise, meaning helping scientists refocus on their expertise, their scientific question, the data, and then helping our teams come together. There was actually something, so just a few takes, well, actually, let's go to our agenda. So as I said, we'll talk a little ChatGPT data. We actually wanted to then introduce you guys to working with Parquet files, Apache Arrow a little bit. Anybody who's already sort of employing Parquet files in part of their workflows or has discovered IBIS, for example. I don't know if this is going to be interesting or novel to folks. If not, that's good. I think that even that as a concept would be useful to you. And then we'll go back to the link as well. But if anybody wants the link, the notebooks will stay available. Okay, so ChatGPT, I guess, just to talk about the advances in generative AI and everything that's been happening. It was really interesting to go to the AWS symposium yesterday, and I'm sure they'll have a bunch of public information about the tools they have available. A lot of them are still through SageMaker, and I'll have Adam talk a little bit about this as well, because I think that this is one of the things that AWS, I guess, in my opinion, really has cornered the market on is making traction in areas where scientists want solutions, right? So if the scientific community is wanting some sort of application of LLMs, they're going to give you some way to deploy LLMs in AWS, which I really appreciate as a service. I think the actual deployment and getting past that first step is still very tricky. But what I wanted to say is that their tools are coming out. And so as a community, when we think about deploying some of these models, whether open source models or if we want to sort of, train them on our data, the tooling is on its way, but it's sort of not quite there yet. But there are really interesting things that they said that I wanted us to take away. And I actually just have my notes here. So the one thing that they said yesterday that I thought everybody honed home was that you still have to formulate your problem correctly. You still have to formulate your question correctly. And the most important thing I think they said is that no AI solution is going to get us out of the dilemma of dealing with our data, unfortunately. So that is sort of the, if anybody needs a reason to advocate for managing your data correctly back to, whoever your team is, I would have to say that even the folks who are trying to hype this up as much as they can and sell the solution are still telling you that this is only going to, and I think we have a really good slide that Jordan put together, that this technology is only going to exponentially, what did we say, your data?
Well, it's only going to get harder and harder to really wrangle this up. So
Right,
I would say
right.
that really helps. And with big data, something like a Parquet file is really nice because you can partition it, you can distribute it and access it in ways that you really can't do with a CSV file.
Right. And so folks were telling us that you still have to ask the right questions. And a lot of this we covered in our sort of organizational aspects. You have to ask the right questions. You still have to have a domain expert present. There was one company who was actually a startup. Was it Recursion? No, it was Regeneron. No, it was Regeneron who was saying to pair. Oh, okay, sure. Small. Okay, yeah. So Regeneron was actually saying that what they actually do is they actually take their domain experts. So they're a drug design company. Did I get that right? Yeah. They're a drug design company, and they actually take their medicinal chemist, and they actually have them sit with their data scientist as a full-time job. And they actually sit together and they pair code together because it is that important to them that the two sides of this equation are aligned. And they said that sometimes that's a big ego hit for a medicinal chemist who is used to being in charge and who's used to calling the shots and having his intuition lead the way. It's a really big deal for them to take a step back, to sit in a room, to code with somebody. But that's the only way that they've actually seen progress being made, because they need the data scientists to get real life feedback as they go, why the predictions that the model is predicting are going to be different, for example, than what the medicinal chemist would recommend. And the model needs to get that real life feedback. And so that collaboration, I think we talk about as well, folks were really emphasizing. Let's see. Some of the things that I think were hyped, let's say, things that people were saying that I think are not going to be true, is the concept of outsourcing drug design to algorithms. In my opinion, and it's just an opinion, I'm kind of curious if anybody has a similar opinion, we will never leave AI alone to do drug design. Is anybody on a different path? Is anyone trying to create something? I'm just curious. Does anyone have high hopes of sort of, there's companies forming now that are essentially run by AI. So it's like a one-person company and then AI does the rest. I think that will never happen in drug design, just an opinion. And the other good thing I would have to say is that, so let's see where I have. So the three things, I'll go through some quotes that I heard. I would say that the best thing that I heard from the conference yesterday was that essentially it sanity checked where I think the field is going with these kinds of models. And so you guys have to let me know if you think we are in the same three options. So essentially AWS and its partners that came to talk to us told us that there's three ways that we can use essentially large language models and generative AI. One is to use it as it is. For example, text editing. I don't know if it's legal in anyone's company, so you don't have to raise your hand that you've done it. It's not legal at BioTeam. I've done it. but using it essentially to homogenize text, right? So if 3 scientists are writing text together, you can use things like ChatGPT to actually homogenize the text and make it sound like one person wrote it. Anybody else sort of poke around and use it for general text editing features like, I don't know, elaborate on this sentence? No? Yes? Yeah? And what kind of thoughts do you have? Right. You got to do some prompt engineering, right? So, okay, but also in the sense that, like, let's say you can also say, I am a scientist writing a scientific paper, and I would like for all of this text to sound like one person wrote it. It will understand that as a concept, and it will rewrite it for you. But you do have to do some prompt engineering, right? Right. So I know that Stan, for example, here at BioTeam, he loves ChatGPT for fun things. He's trained it to be a Socrates teacher. He has an article coming out at Medium on how to play with ChatGPT. So there is sort of like a fundamental text feature to it, right, about text. Now that is just about the sound of text, because of course it's a model that's just statistically trained to produce an average or agreed upon sound to text, meaning the words that it places one after the other are meant to sound pretty good, right? Because it's finding a statistical calculation of how that text is placed. And so in science, that can work. Now, no one's going to guarantee you that the actual facts that it's statistically aligning for you are going to be correct, right? So we've all heard that models hallucinate. Yes? Okay. But they hallucinate sometimes in really sort of in ways that we as scientists don't let's see, like that we wouldn't think about, right? So they hallucinate on things that should be structured data, for example. So if you just go into ChatGPT and ask it to query something, like if you give it a gene symbol and say, what is the full name of this gene symbol, it actually won't give you the correct answer, which seems like it should be super simple, right? It just needs to go into a structured database. There's a lot of structured scientific databases and output a structured output, right? So it hallucinates in ways that as a scientist, I think you might not be prepared for. Or for example, if you say, so I worked on spinocerebellar ataxias, right? So I worked on movement disorders. If I went in and said, give me the 3 most recent publications on ataxias, right? It's going to not only not give me the actual publications, it's going to make something up. And if it knows who I am, for example, it'll give me publications that I wrote, well, I didn't actually write, but that it hallucinated that I wrote, right? And this is because it's not actually trying to be correct. It's just trying to give you something that sounds statistically probable in the way that it was trained. And so there's really specific ways that as a technology sort of out of the box, it can be useful. For example, sometimes I need idea generation. And so I wanted to hallucinate. I wanted to come up with some nonsense that I can sort of like filter down myself and say, okay, this could be interesting, right? So I will tell it, hey, tell me something about, you know, I don't know, novel AI ML use cases, for example, in biomedicine, right? And then it'll come up with some things that are, you know, it doesn't really matter that it's not true because I have the sort of expertise to pare it down to its knowledge. Okay, so that's one way. So there is a way to use it in a chat-like way. And AWS is also releasing features that allow you to, for example, to chat with your Health Lake data. So I don't know how many people have worked with Health Lake, but they're also having chatbots for Health Lake data. And the way that they're currently trying to get around the fact that there's sort of this hallucination factor is they're trying to give you an explanation or trying to provide an explanation for how that model, how it connected the node to give you the answer, right? And it's an interesting concept. It's a very important concept. And so we are on the way towards trying to get these models to give us some feedback of how they got to the answer. But I would say that we're not quite there yet. We're sort of trying to map these nodes of explainability, but we haven't quite gotten there yet.
Can I add a comment to that?
Yeah.
Sure. So I think some of the problems here is that there's really no one model to rule them all. In these cases, when ChatGPT isn't really giving accurate information, one of the reasons why is because it's trained on a glut of information. If you really want to use it in a way that's useful for biomedical sciences, it needs to be specialized, which means you still need to have well-curated data. So you can't get away from that topic, I think, you know? Like, so that's just something I want to point out.
Yeah. I know it's not sexy, and we've been talking about data for so long, right? Like, I think people are sort of like, they'd drown out the concept of talking about data. They'd rather talk about a large language model that you put on top of it, and then it can just query your data for you. But I think that's not how it works. And also, I don't know if anyone's heard of like emergent properties of these models, meaning that they're sort of like growing faster than expected and they're going to become more capable than expected. That's actually not correct. It all depends on the metrics you apply. And so there's some interesting publications where people actually go into the science of why these models are actually just sort of tuned by metrics. They're not emergent, and their capabilities are also not emergent. In case you want to calm down some of your fellow friends who are thinking that they're about to step out of the box and take over the world. They are not. And these are just really interesting quotes that I sort of gathered from fellow folks in the field, because I think right before we came to the conference, I was like, I don't know how to talk about something that is changing underneath our feet, literally from yesterday to today. And so I gathered some community input. And so, of course, there's sort of teams where there's an expert on board who is going to be able to take advantage of these advancements very quickly. But to think that teams are just going to jump into any of these things sort of without advanced AI/ML teams is unlikely. Would you agree, Jordan?
I agree.
Yeah. And that's sort of like a general opinion. And I think that is fine. And we don't need to turn every company. And I see this a lot. And I don't know how large you guys' companies are. Maybe all of them don't have the option of becoming AI companies. But what we see a lot in BioTeam is that companies essentially come to us, and what they really want is to become AI companies, right? They want to become companies who produce novel AI/ML products. And we try to tell them, you are not an AI company. And if you want to become an AI leader or an AI company, that is going to take a whole pivot of your whole organization, right? To utilize an AI/ML product is one thing, which is what we're trying to do here in the workshop. But to become a leader and an innovator and to design things is a whole other concept. So Fernanda, I don't know if anybody knows Fernanda. She was supposed to be here with us again this year. She was here with us a few years ago. But the fact that ChatGPT is going to give you novel answers, she says, get that out of your head. She has a great article that she labeled ChatGPT as a mirror. And the fact that it essentially gives you back very average answers at best. Because that's what statistically it should do. It should give you something that is statistically reasonable. And she thinks, or that's her statement, and I semi-agree with it. I think there are ways to tune it to biomedicine, but there's no such concept as make this small change and all of a sudden it's going to be useful for biomedicine. And in fact, I think I'll show you something later where they actually assess different models for capabilities of predicting on genetics. And the general models like just ChatGPT are actually better than some of the BioGPT trained models. All to say that just training in a biological data. And also, I actually missed this whole Galactica thing, that Meta released Galactica. Did anybody follow that whole up and down? No, I also somehow missed it. They just recently, everyone knows who Meta is, right? So they released a model that they said was trained exclusively on biomedical data, text, scientific literature. It was actually meant to guide scientists through reading articles, right? That's what a lot of us are imagining. As scientists, we all have to sift through articles and we're like, AI is just going to tell us the summary. Anyway, that was the goal of Galactica. And apparently all it did was just provide a ton of misinformation, and they shut it down. In fact, they shut it down before I think any one of us even realized it was out because of the huge amount of misinformation that it was producing. So if anyone's interested on how that worked out, you can read up on Galactica. Okay, so the features that we went through. All right, so if you just want to use it in a ChatGPT way, you have to think about prompt suggestions. You can give context. There's a lot of sort of fun articles on prompt engineering. I think it is fun to play around just to sort of see what it comes up with, right? So, you know, as a scientist, I want to, I don't know, write an email to IT asking for, you know, more compute or something. Make it sound very convincing and sort of, you know, irresistible, and just see what it comes up with. To me, that's fun. But in terms of science, again, think about using it to just smooth out some text, things like that. I don't know about the legality of anybody's companies, so there's a whole legal feature involved. Right. Okay, so then the next step, as AWS presented it and sort of as the community agrees to it, is creating your own interface to a large language model, whether or not it's an open source large language model or if it's OpenAI's large language model, right? So in this sense, you are essentially putting in you can tell it what information it needs to look through. For example, you can put in some structured databases, and this is actually where it can start giving you appropriate results from structured data. So if you pointed at a table of gene IDs, for example, you can't actually ask it, what is the name of gene ID 27? And it will actually give you the correct answer because that is information that you sort of fed or pointed it to. So if you train it on structured data, and what did we learn, Adam? Who was training it on structured data? Do you remember from yesterday? Somebody was specifically saying, I don't know. Anyway, but so there are teams that are trying to input more structured data into these models so that it actually learns how to read out tabular data or columns or spreadsheets and things like that. Because at the moment, that's not what it's trained on. It's trained on unstructured data. And so there are ways that are manageable to actually sort of pivot the lens of these models to some structured data that you possess and then query it in some way that really, really reduces sort of the hallucination factor. And that is possible. And again, AWS released some options for how they allow you to do that through SageMaker. You have access to these open models that you can sort of directly click and implement. And then the hardest way to do this is to actually sort of set up these models internally. The other thing I was going to say is that companies like AWS, the other benefit, and again, I'm not trying to sell them, just nice to see what tools are coming out, other companies will follow. The other interesting thing is that they are interested on keeping your data safe. So they are trying to work on ways to allow you to point this at your own internal data and keep your data safe, because at the moment, the actual reality is that these models then suck up whatever data you give it, right? So if you give it a bunch of your internal data and ask it to query it, it's going to actually pull it into the model, and so it becomes public data. And so companies like AWS are trying to find ways to make your data secure and allow you to query it through these ways. And then again, if you want to become a leader in this specific field for whatever reason, then you're really going to have to deploy these models internally, which is a whole other capability play in terms of the quantity of compute you're going to need, the internal setup you're going to need. You are no longer just sort of point and clicking at random teams. You're going to deploy these models fully internally for whatever capability. And again, people kept reiterating that you have to have the right question. Okay, and the other thing that I wanted to point to, okay, so before we get into sort of like ways to assess, let's say, some of these models, because I just have, I think, a few publications or maybe even just one. Any questions on that? Or Jordan, is there anything else you wanted to say about that? Adam? No? Any questions or comments? Anyone who's tried any of this? Okay. Okay, so then the second portion we have to get to is essentially, how do we know whether or not these models are going to give us a bunch of nonsense, right? So if it's just you as a scientist playing around with it, that's great. But as a company, as an organization, you have to have some way, let's say you're even training on internal data, you have to have some benchmarks. And Jordan had said this best when we were talking about this. His favorite part is that people are actually doing some analysis or some benchmarking and not just sort of going out and doing whatever. And so this is one example of how you can do benchmarking, for example, for genetics. I don't know how many people are in genetics or do some amount of genetics, probably at least some of you, right? Anybody know what genes are? Okay. So anyway, well, it could be that all of y'all are on protein prediction, and protein prediction is a whole other ball game. But anyway, let's talk genetics. So as you can see here, they have sort of like the different categories of questions, right? And which one did we talk about? Okay, so, right. So SNP associate, okay, here we go. Convert an ID to the official gene symbol, or give it, or let me know what it actually is. So you can see all the funny ways that as a scientist, you wouldn't necessarily expect a model that is just looking at tabular data to give you false answers. So it's going to give you can see here the score. So it's sort of giving you not a wrong answer, but it's also not the right answer, but you would probably know that it's the wrong answer. it's giving you a correct answer or it's giving you completely the wrong answer. And if you play around with it, you will see that you can ask it a totally fake gene symbol. You could say, tell me the diseases associated with, NSO at 12, and it will treat it like a gene, and it will just make up things and potential cures and research publications. And so it does have the ability to just sort of randomly assign gene symbols and names. Obviously the same for SNP locations, which are even more sort of like scientifically rigorous, and you have to understand what you're doing. And when you get all the way down to sequence alignment, for example, it essentially knows that it can't do it. It's like, you are asking the wrong person. I am just an AI model. And so it's even going to have some sort of coherence about the fact that it can't do it. But that is actually your best case scenario, right? So when it tells you that it's not capable of producing this information. And so if you look at, this is obviously from the publication and you guys can go read it. So whether or not it understood the question correctly at all. And so again, what you can see is here we have BioGPT, and then we have New Bing, which I actually, this publication makes New Bing look really good. I don't know. We've played around with it, and I would say that like I prefer ChatGPT over, I don't know. It's anyway, don't take this as an advertisement for New Bing, I guess is what I'm trying to say. But I think it is important that, for example, BioGPT didn't even really sort of like understand all the questions, although it was trained on biomedical data, right? So it didn't really even get the thing that you're trying to scientifically ask it. And then whether or not it actually understood that it can't answer the question, right? So incapacity awareness is it didn't know that it's not trained to answer this kind of question. And sometimes it was. So again, for this sort of DNA alignment, it sort of universally said that it can't do it. And then how often did it hallucinate and the overall score? And so you can see that the biomedically trained models are not giving you much of a score, and certainly not a score that's better than a general model. And so if you want to have any kind of success with these kinds of technologies, I guess the point is you're going to have good data that you train this model on internally. Then you're going to have domain experts that are sitting there and benchmarking it, right? In a similar fashion, something like this, essentially. And you have to know whether or not the data that you're perpetually feeding into it is improving your metrics, if those metrics are doing anything for anybody, and then it becomes something that teams can use. And there are different very cool ways that teams that are actually like AI research teams are using this technology. So there was actually a company called Generate Biomedicine that was talking about ways that they are using this technology to actually better predict protein functions, to better predict clinical trials. There's a lot of talks at BioIT that I also benchmarked about clinical trial patient selection, about protein synthesis. I don't know how much they'll actually tell us about what they're doing, but there are research teams that are actually making progress on this. They are actually putting together these modules of LLMs, getting them to think about toxicity and immune response in ways that they haven't before, synthesizing sort of patient cohorts. All of this is happening, again, but by teams who are able to be experts, pick up this technology and go. Let's see. Okay. So the conclusion on ChatGPT, and then we can sort of take a break and pivot into some data, that it does still make very much domain-specific mistakes, right? And that is really important to consider. And open source efforts are on the way. Hugging Face, Bloom.
Yeah, that's good. Yeah.
Yeah.
Hugging Face probably is one of the ones.
Is probably the best one. Right?
I've used some of the models.
So I would say, I guess, don't sue me if you get a publication that is cited for this. I would consider a general knowledge search is interesting to me. Summarization, refactoring text. It can start helping you in your bioinformatics pipelines. And I think that is a real way to use it. And especially if you have a bioinformatician that has domain expertise. If you have a bioinformatician who's using ChatGPT because he doesn't know anything or she doesn't know anything, well, if it's a she would know everything, then it's not going to work. And you can also use it to translate. So there are, I think, going to be ways to use it on top of knowledge graphs. Has anyone heard of a researcher that from Birmingham. So Matt Might, has anyone heard of Matt Might? He runs a precision medicine initiative. And he actually had a son that was diagnosed with a really rare disorder. And he, as a computer scientist, actually became sort of a biomedical researcher. And so he helps teams find knowledge graph associations for rare disease. And so as someone who's a computer scientist, he is deploying, for example, these models on top of his knowledge graphs to query the data that they already have sort of synthesized inside knowledge graphs, right? But that is really advanced. And sort of his specific passion and expertise, I would say, but it is possible. Adam and I were arguing yesterday about long chain and that it's not just there for chains, but it's essentially a framework. Frameworks are coming out that are going to make these things a lot easier to use and are going to allow you to use some of the capabilities, like putting together different concepts into a sort of one model. And so as they come out, I think as a community, we have to keep talking about them to realize if anyone's actually having any success. Okay, I actually have a few ethics slides, but I feel like you guys are tired of me talking. So why don't we skip over to Jordan and do a little bit of data and run some notebooks, and then we can come back to ethics, maybe like halfway through or something. I have an ethics framework for you guys. Wait a second. Oh, I don't know how that happened, but I flipped it. Okay. All right. Jordan, you want to?
Well, yeah, the introduction part, if you just want to introduce the demo.
Oh, yeah. Okay. Okay. So an introduction to the demo, and I think you guys have the link. And so our actual, I guess, to give you a background of where we started. So the place we started is that as a scientist, I remember reading a publication and holding it in my hands and then saying, I want to reproduce some part of this publication for my own research, right? And I think that happens with AI ML use cases as well. And sometimes this singular publication is the only ounce of hope that we have that this technology or this thing is possible, right? That someone used some network to analyze my kind of data, H&E stains, or brain slices, or mitochondrial stains, or whatever it was, right? Like someone's going to save me from the dread of counting mitochondria. And then you take this paper and then you take it to someone who you hope can help you deploy this model on your own data or train it. And it sounds like it should be possible. And Jordan and I started off this workshop thinking some aspect of that should be possible. There is some way to take something that's published and help a team actually make it happen for themselves, right? But then what we very quickly found out is that, so we wanted to start off with a publication. What we wanted to do was actually ask everybody to break out into teams, find a publication. We would try to get a publication and sort of like reproduce it, right? What we found out is that, first of all, we will never get the data from any publication. And the reasons are numerous and wide and incredibly frustrating. And if no one has asked you from the scientific community before to actually publish your data in a way that someone can reuse, there's a FAIR symposium next door. Please feel free to think about that as a concept. Anyway, it became completely impossible. And nothing that we could reproduce for you here. So we went on the search for open source databases where we could actually use the data to do something fun for you. And we ended up with Open Targets. I don't know how many people have used Open Targets before. Okay, a few. Nice. Great. That's very good. So it's not sort of going to be boring to everybody. But I think it's a really great framework for how our internal data should be structured as well if you want to run any of these visualizations on it. And so this is sort of where we landed. We'd love some feedback from everybody after the workshop if this ended up being sort of not technical enough, too technical, interesting, not interesting, because we do want it to actually be useful as we iterate on the workshop. So.
Yeah, and just to add to some of the frustrations I've experienced going through publications, looking for good data, one of the things you'll commonly notice is that they'll have it just camera ready, right? So it will serve a specific purpose. And you might be able to reproduce it, but then how do you adapt it to your own research? How do you use it for other things? And when it's basically something like a data set that's just maybe just have some labels, but no like intuition behind what you would use it for, then it's like, oh boy, I got an accuracy of 95% on my model. What now? So it's really hard to connect interesting research questions to a lot of the data that's out there. So I had to go on a very long journey, I feel, to get to something like open targets before I was like, finally, something connected. Because for me, coming from from information retrieval, I was kind of spoiled with like Wikipedia and other things because you can extract so much information from that. But that's not really quite the case for a lot of the biomedical data out there. So that's really what's motivating this. Yeah, so I did try. Those are good things, and I really support that, especially for publications moving forward. It really is, I think, making a difference in terms of being able to host and share data. But what I would love to see too is connecting it, kind of like in a platform, because it's still hard to be like, well, I like your data over here, I like your data over there. Can we combine them somehow? Can we use them together? That's a big pain right now.
Do they have any references in that Figshare data to some sort of global IDs that you can then pull other information from, right? So, or is it just a closed data? I guess like a lot of it is just a closed data set. Right. Right, right.
I think the hope is that maybe AI is going to swoop in and fix it, but I'm not sure. Yes, that was definitely one of the places that I looked for data to try and grab data out of. So I still support that definitely, but I'm focusing on open targets. I think mostly because
there's some cool features also, like the Parquet export.
Oh yeah, so I'll definitely get into.
We'll get to it. We'll get to it. Okay.
But before that, we have the link available here, and I will be going through the demo. But I just want to cover some of the reasons why I was exploring this and why I think it would be useful for you to take a look at some of the libraries I was using. Oh, sure, yeah. Wow, they made it idiot-proof, and I still can't use it. Oh, the big green one. Okay, so there's two green ones. Maybe they should have thought about this. All right, here we go. So I'm just going to introduce open targets really fast.
Again, not for the sake of selling open targets, but for the sake of pointing out features that make a really great, think to yourself, internal database, right? So think to yourself, whatever your teams are doing, how can I translate these features to internal databases that then make my data have value past its experiment? And for it to have value past its experiment, it needs to have some specific features.
Yeah, and I would say that my other experience was something like a good platform like NCBI. If any of you have been there before, a lot of great data are available there. So I was looking for something in that kind of similar vein. So what Open Targets is it's an open source platform that allows or facilitates drug discovery. It combines data from a lot of areas together in a way that allows you to explore it pretty intuitively. It's got a useful web interface that you can search, for instance, for a disease and look for things like, well, what drugs have been used on it? What are likely targets? And a lot of the data can be pulled in also from other sources. So it's just a really great way of synthesizing information. And I was just immediately impressed. But secondly, this is important to me because before I even start making models, I need to understand the data. I need to understand how it gets connected. So I really feel like open targets, and I know I say that a lot. I'm really not selling them, by the way. I was just really relieved to finally have. Oh yeah, big thing there. This is open source. And I'm going to get into this, but you can download the data yourself. You have access to it. So it's a wonderful effort, no charge. You can go on there and grab it or explore it through here. I think it's a great tool to facilitate research, and I can imagine. Oh, certainly. So, I mean, I was using that to kind of drive my model development. Right now, I don't think they have that as a feature on the site. But one thing I want to point out is that this is also an open source platform. So you can download the code and stand up your own server, your web server. You can add on new apps. So there's a lot of potential here to adapt it to suit your own needs, even if you're not into pharmaceuticals. So that's just something I want to point out here. It's just a good model.
I would just also add that a lot of teams that we talk to, they're trying to launch AI/ML initiatives. Their first step is we are going to use open source data and sort of demonstrate the potential value of our AI/ML models, and then we will launch it on internal data. But I have to say that a lot of teams get stuck right there, right, Adam? I see you shaking your head, that even just right there, it becomes impossible because they can't find any examples for just showing a potential proof of concept. And so that's even important. Yeah.
I agree. And I think another issue is that even when you have all that data together, how are you going to get it to your researchers or scientists, right? So that's usually like an afterthought, but in my mind, it should be your primary thought, right? So if you want to drive drug discovery, you need to make really great tools that are interactive, that can really drive that insight. And that's something you just don't get by having a bunch of data, right? So a lot of like love and care was put into this platform in a way that is actually user friendly. And I thought that was really great. And I just, again, just want to plug it because I just love it so much. All right, so moving forward. And this is just an example. I just want to include a quick screenshot of something you can do. I was just looking for like also Alzheimer's disease, what drugs are linked to it. And you can see here that you get this nice big table. You have a lot of like links that link out to different sites. And it's a nice way of aggregating data. But on its own, it can still be difficult because how do you pull this data out? What if you want to use like a machine learning model, right? So this is great for a researcher, but we also have to think about how do we have a layer on top of this to make it like accessible for things like machine learning algorithms. And that's what I'm going to cover in a second, because what's really nice about Open Targets is that they put a lot of effort into aggregating this data and making it available in tables. You can either get it as JSONs, or as Parquet, which I will go into in just a second. I love that format. I had just discovered it. But another thing is that they also have a GraphQL database behind the scenes. So you can also just actively query it and retrieve information on the fly. So there's a lot of endpoints, a lot of ways to access data. And I just think this is a great way to really make your platform more useful in general. So what are these Parquet files? Well, it's a file format that stores columnar data. Sorry, and it's just like a CSV, for instance. You could have like your variables stored in here, but there are a lot of advantages that already come built into this. First of all, it's efficiently stored as binary, and that's just by default, right? As soon as you take something and store it as a Parquet file, you save a lot on space. And also it can easily be partitioned. Why this is useful is that if you have a giant database, you might not necessarily want to load it all at once, or you want it to maybe be distributed, right? So this supports things like multi-threading or distributed processing. So it's just nice to have that as well. And it can store deeply nested data just like JSONs. That's also, well, that's a blessing and a curse, but we'll get into how to actually deal with that in a little bit. So it's not a flat file though, like a CSV. It's got a lot of rich data associated with it. And it can be a lot faster to read, depending on what you're using. Like for instance, Pandas, much faster to read a Parquet file. So that's another thing just to keep in mind, is that if you need to load things up quickly to like serve to someone, sometimes like speed can really matter here. There are already a lot of libraries that are supporting this. Apache Arrow, native to that, it's already supported. DuckDB and IBIS are the tools I'm using. And I'll go into that a little bit later too. And I've also included a link here too if you want to go ahead and take a look at it at some point. I'm sure we'll make the slides available later, so I'll just probably zoom.
If you have any questions about it, we'll be at the booth if you guys just want to chat. Like,
And what's also really nice about this is there are schema associated with it. And if you worked with databases before, you might be familiar with this concept in the sense that, like, for instance, blood pressure, that's going to be something like an integer, no, a float really, right? So it's really helpful to know what kinds of data you're looking at a glance. If it looks something like at a CSV, I'm like, well, what am I looking at? What data types, you know? But with this, with a schema, not only do you have that information at your hands, but metadata can be actually included in this. So you can explain like, well, it's in these units, or we followed these protocol, or the author of this data set is, and that's a great thing to have in there too if you put all the effort into it.
And that's one of the things, actually, the context around the data is one of the things that we really struggled with, because even when we could get large data sets from publications or from somewhere else, they usually lack the majority of the context around that data. So it was impossible to tell what you're really training on, what this table includes, anything about it that would actually allow you to understand whether or not your training of the model is giving you any valuable results, because you're essentially just in a black hole of data points that mean nothing to you. And so context is so, so valuable, especially if you want your data to be reusable by anybody else except for the person who generated it, right? Yeah.
It's an excellent point. I think that actually summarizes it beautifully. So, and so in general, yes, Parquet files have rich schema, and I would say go check them out. Now, another topic I want to talk about is in-memory processing, because as we get larger and larger data sets, one of the things that we run into is it's actually really hard to do a lot of data wrangling with it. Like for instance, I used to use R a lot, and so I would just like to put everything in my data frame. And that gets very difficult when you're dealing with lots of databases and trying to join them. It's like, good luck. So, well, SQL is useful. It's hard writing out those long strings, in my opinion. There's a lot of support for it, yeah, but I'm just looking for something that hits that sweet spot. So what I'm advocating here essentially is that we need to think about things that can work or get around the fact that we can't have it all in one spot at once, right? Like you don't necessarily want to have your, I don't know, 100 gigabyte data set loaded into your computer, right? So we want to look for things that support lazy execution. I'm a lazy person by default, so like I like that term. So, but what lazy execution means in this case is that you only run the stuff you really need. Like even if you call something over all the columns, you know, if you're filtering stuff, it doesn't actually call on everything except the stuff you select or filter for. So it allows you to be extremely efficient and only pull and use what you need and not have to do it over the entire data set. It's great for rapid prototyping. It means you don't have to worry about having the entire data set to explore it. And the tools that I'm going to use that support this are IBIS and DuckDB, which I will get into probably right now. Oh, yeah, oops, went too fast. IBIS, okay. So this is a great tool. It's in Python, and it's a native interface that supports up to 18 data backends. And the idea is that, you know, there's a lot of different languages and a lot of different ways to access the data, but could we perhaps unify it, right? Could we create a simpler way, maybe a language to specify a query without having to know what the backend is? You know, this can really speed up development. This can make things easier. And for people who have used things like dplyr, for instance, it's very familiar in the sense of how you can set it up. Here I have an example here, Make Your Codebase Portable. Provided by Fernanda. She's not here today, unfortunately, but it was really nice of her.
Yeah, so actually just to say, just to also say how we always all are learning. So Jordan was initially working with DuckDB, right? And then Fernanda, who is actually, I think she has a whole company that is essentially committed to getting people to use more Parquet files, but she introduced Jordan to IBIS. It's open source, right?
Yep.
Yeah. And so because it was so fantastic, he pivoted everything we did into IBIS in the last, I don't know, couple days or whatever. The time span doesn't matter. The point being that we are all always iterating. And so whenever you see your teams sort of, you know, pivot and take a left turn, sharp left turn somewhere, sometimes it's fine. Sometimes it's not. And sometimes we have to sort of like stay the course. But all to say that new tools are always coming out and it is possible to pivot.
Yeah, and for me, what really drove it too is that there was a lot of problems with just doing a bunch of chain SQL queries. There wasn't a lot of optimization in the backend, and maybe it's also because I'm kind of terrible at writing those queries. I don't know, but when I switched over to IBIS, it really helped. And again, I'm not trying to plug these things. It's just such a revelation when you finally get some good tools, right? So when I switched over, it just really enabled a lot of things, and that's what I want to kind of highlight here. Is that like this is an example of code where if you've ever used something like dplyr before, it might look familiar, where we're doing a filter, then we're grouping by some values, we're aggregating that information and sorting it. So you're chaining them together like a pipeline, very intuitive, but then it gets translated into like SQL or any other kind of, it's a backend. So you could be doing it locally and then send it to the cloud or have another kind of platform where it actually gets run, and it doesn't matter what the backend is, you just switch it over. So I think that's a really great way of like standardizing development, and I fully support it.
And if you guys decide to take this back and try it you yourself, or you want your teams to try it, you can always email us and we can try to help you navigate it.
And DuckDB, I'm not going to go too much into, but essentially it's a way of providing a unified API interface across different kinds of files. It's kind of cool because, like, for instance, I could read a pandas data frame in memory as if it were like a database. And what's really nice about IBIS is that DuckDB can be a backend, which means now I can use IBIS to query the data frame. So, well, that's a little nerdy stuff. It doesn't necessarily matter more so than the fact that it's really useful for development. It's already supported by many languages, so you probably won't have any issues to find the right one. Yeah, I would say it's part of also a growing ecosystem of Aero native tools. Has anyone heard of the Aero ecosystem at all? No, well, I was discovering as I was going, and I think that it's definitely up and coming. And I'll talk about it a little bit, but essentially, these are the kinds of tools that you would maybe want to start exploring if you're thinking about maybe doing like distributed, like machine learning algorithms or data processing. This is something to start checking out, I would say. Right. Okay. Yeah. So I do have a slide on this, but just the quick intuition behind this is that there's just so many different ways to like serialize your data and communicate between services. It can be a bit of a pain. So part of the intuition behind like the whole Apache Arrow ecosystem is like, can we just pick a standard already? Right. So that was the idea, and there's a lot of support behind it, a lot of services, and I think it's great, and I am continuing to explore it. I'm going on a journey, I would say, and I would say there are a lot of advantages to using it for your data layer. Any comments? That's good. Good, that covers it. Great.
Okay, so let's go through.
Yep. So we're putting it together. We're going to now go through those tutorials. Hopefully, I have quite a few visualizations, so hopefully the computer I'm going to use doesn't choke on it, but we'll see. So let's head over to the laptop, I guess.
Yeah, I think you probably have to go over there.
Yeah.
Mhm.
Great.
Anybody need a stretch or a break, or are we good to keep rolling? What do you guys feel like? We have, yeah, we're 1 hour and, oh, perfect. Okay, so should we take like a 5-minute break before we start with this stuff? Okay, yeah.
Good idea.
And then, yeah, you guys can just stand up and get some blood flow going.
Good so far?
Great.
But come back.
Yeah, start again.
Yes, please.
Less than 5 minutes. 9 o'clock.
Well, ish.
It's fine.
This is not. Yeah, you're doing great.
You're a generative text machine. I don't even.
Why?
Because you just go.
Talk.
Go. Right? Isn't it kind of like amazing? You're not even looking at slide.
Yeah, it's like you just didn't even have to prep. You were just like, it's great.
Just wind her up and she just goes.
I'm a
I
generative.
love this. You are.
It worked. I liked it.
Generating language. Yeah. Don't mind if I do. Like the 1 second you like, Adam, anything? Nope. Okay.
I'm so inclusive.
Anyone else need that? No. Okay.
What for me? Oh, great.
I'll keep going. Continue.
Yeah.
Is it compatible to Snowflake database?
Arrow?
Ibis?
Or
Yes.
Ibis? Oh, yeah, yeah, for sure it is. Yeah, a lot of people are using Snowflake, yeah.
And then you can store the genomic and the molecular information in Snowflake database. Like there's a way you are storing in WB.
So I was actually just reading it from the Parquet files directly, but it does support Snowflake. So like in practice, yeah, you should probably be storing a database for these notebooks. I was just taking it from my laptop, but I got it from Open Targets.
Yeah, and Ibis is open source, right?
Yeah.
Yes. All the things we're exploring are open source.
So how is it different from normal NCBI, querying a gene or a protein?
Well, I think first of all, it's like having to remember all those APIs is kind of like annoying, right? So it really, I think, combines a lot of the data in a way that's queryable. So I can join tables and search for things in a unified way compared to NCBI. And there's many differences still because it's not necessarily storing the nucleotides or protein.
Is it what time again? All right, looks like people are coming back in. All right, Jordan, I think you're gonna have to sit over there with his, yeah.
Before I do that, though, I just want to go over quickly just what we're going to be covering, hopefully if we have enough time. I have 4 notebooks here, and I just wanted to describe some of the highlights about what we'll be doing. I'm going to keep it high level, though, like, so when we're going through the notebooks, I'm not going to walk you through every line, mostly because that's a little bit boring, but also because, you know, I just want to hit on how this is making it possible, because that alone is actually really terrific. In the first part, what we're going to be doing is exploring the molecules data set from open targets. These are small molecules that are drugs. And first of all, I'm going to use IBIS to ingest that data. I'm going to use these SMILE strings. If you're not familiar with them before, I'll talk about them briefly. We're going to use that to build a similarity matrix between the molecules based on how similar they are in their composition. We're going to cluster them. If you're familiar with like dendrograms and stuff, we're going to be able to actually visualize that. We're going to embed using this thing called UMAP, which I'll expand more in a different notebook, and obviously lots of visualization.
And just a really quick note, Jordan did this work. Jordan pulled together these different notebooks, sort of exploring the data. We are not, we don't design small molecules. So the idea isn't to tell you the kinds of visualizations that your scientists or you, for example, would want to do. It's only just to say that once the data is in a particular format and structure, you can do a lot of things, these being some of them, but these are not specifically the visualizations we're recommending, for example, right? So it's just to say that now there's a whole host of sort of easy reaches once your data looks like this.
Yeah, that's a great point. Like, I'm definitely not going to advocate for that little model I'm going to show you later on. Like, don't predict using that thing, you know, but I just wanted to show what's possible, right? So yeah, I think that sums it up nicely. Part 2 is when we're going to start exploring diseases that are in the database, like Alzheimer's, like I mentioned before, that's one of them. So we're going to grab them all, and we're going to figure out ways of vectorizing gene expression associated with those diseases and using that to represent them. And that's a complicated concept, so we're going to really pass to the visualizations to see what I'm talking about. But also we're going to use the descriptions, right? So there's like free text associated with each disease that describes it. Well, that's useful, and it's about time we start using it, right? So I'll show you how to tokenize it, how to use it for embeddings. And these embeddings aren't just great for visualizations, they're features that you can use downstream for your machine learning models. So that's part of the reason why we're exploring it. And of course, more visualizations like the one over here. On part 3, what we're going to do is basically something very similar for the genetic data. In particular, we're going to be exploring targets that are protein coding. We're going to query open targets for these IDs that are ensemble IDs, and then I'm going to show how you can actually pull in the transcripts from ensemble, the database, and then use it to actually to embed as well. Like, you can take the nucleotides. If everyone's ever heard of K-mers before, we're going to use that to vectorize it. Same thing with the translated proteins. So a lot of options here. Same thing with the gene descriptions and Go terms, if everyone's heard of that before. That's another option. So I'm just showing you all kinds of ways in which you can explore your data after embedding it, plus visualization. And then the final part, if we hopefully have enough time, is we're going to try to use these embeddings together in a way to train a model, for instance, and then use these association scores that are in open targets. And what those are essentially is that it's like if there's any evidence, let's say experimental evidence or evidence in like publications, or maybe from like associated pathways, like genomic ones, that can be used to infer that maybe a disease and a particular target are linked in some way. And that's useful information if you want to start exploring drugs that might target it. But again, even if you're not into things like drug design, this is something that's useful, I would say, for other purposes. And then I'm going to show you how to visualize it, because what's really cool here is it's almost like a heat map is what I'm showing you. I'm showing you an embedding of targets, and I'm like, what ones are associated with Alzheimer's? And you can see right there, like the heat map, the ones that are like yellow or reddish, those are maybe ones that have a higher likelihood of being associated. Now, again, I just want to stress, this was just a dumb little model I was using, so I wouldn't go and run and start, working with this, but it just shows the potential of connecting a lot of the insights of a model to visualizations. So without much ado, let's go ahead and go back to the link. If you haven't followed it already, go ahead and click on it. And if you have any issues, just let me know. But what we're going to do is actually, do we even need this yet? Or?
No, I don't. No, you don't need that. And then Adam and I can be maybe here. I don't know what value can we add? Maybe no value. Yeah, go ahead.
Sure. Thank you. Okay. All right, so I'm going to try out this link, and you can, oops, I already messed something up. There you go. Oh, duh.
This isn't Jordan's laptop, so we also don't have any experience on whether or
Oh.
not the laptop's going to be able to carry this, but we'll see.
This one? Oh, and then how do I?
Oh, he's got it. He already
Right
opened
click
it.
or- Okay, so now everyone can, oh yeah, good point. Okay, great. So hopefully everyone can see this just fine. I'm going to load this up and hopefully the visualizations won't crash this thing. Oh, okay, that's a good sign. We're now entering the notebook. And the goal here, like I was saying before, is that for the first one, we're going to explore some chemicals, some molecules. Well, they're chemicals technically. And we're going to see how we can pull that out of the database from open targets. And before that too, I'm just going to demonstrate why I love IBIS so much. And we're going to try finally embedding these molecules in interesting ways and running some visualizations. So this is an example where I'm just, ahead of time what I did was I just downloaded the targets database from open targets. And now I access it. It's a Parquet database. So it's this directory with a bunch of partition files like I was saying before. So that's a good thing. And I just read it in real quick like this. What's really cool about this too, because this is notebook mode, is you can, oh, I hope this will respond. Can it? Uh-oh, it's not like, okay, I can use my little arrows here. Because like it's an interactive notebook, so you can go ahead and play with it if you want as you go along. In this case, it's just showing all the fields in the table, kind of like the screenshot I showed you before, but now we actually have access to it, which is really great if you want to perform some like data transformations. Oops. So why would you want to? Well, here's an example, subcellular location. For these drug targets, particularly like for protein coding ones, they could be expressed in the cell in particular locations. These are useful labels. You can use them for coloring graphs, like what I'll show you later. You could also use them to make predictions, but unfortunately, they're deeply nested. What this says right here, again, this is a schema that we're seeing here of data type, is it's an array of these structs. And these are things that are like dictionaries, if you ever like messed around with Python before, things that have fields and associated values. So for instance, this has location, which is a string, source, which is a string, etc. But how do I get that stuff out? How do I do it in a way that's scalable? Right now, it's so deeply nested, all you see are just these curly brackets. Well, using IBIS, what I can do is, first of all, start by unnesting it. This allows you to take that array, and for each element, you transform it into a new table where each new row is going to represent an element of the array. So already we're in better shape, but we can go further by using this thing called unpack. And what this does is that struct field, the thing that was like all in one column, we're now going to unpack it so that each of those fields is now its own column. And you really want something to be in this shape when you finally get down to it, because a lot of your processing is going to rely on columnar formats. And usually it's not very happy with deeply nested data. So I just wanted to demonstrate how powerful it is. Just by a couple of quick transformations, we already have it in good shape. Now let's move on to actually exploring those molecules I was talking about. This is yet another database, and I just downloaded it as well, and I'm now loading up as its own table. Again, though, I'm not loading it all up, right? So this is lazy evaluation. You're only seeing in interaction mode, you're only seeing the first 10, right? It's not going to go and grab up all of them, right? We don't want to do expensive computations on things that are very large. This data set isn't so big, so it wouldn't be a pain, but if you had like millions and millions of rows, then you'd start seeing why you'd want to do this. Again, if you want, you can explore it. It's there, you can just scroll around, but I'm going to start going down, also showing another table, because again, this is one of the powerful things about IBIS, or just treating things like tables, is you can start joining them. This thing is called mechanism of action, and what it is a separate table where it associates one or more of these chemical IDs, that the molecules that we were looking up at here, with an action type, like is an antagonist, is an inhibitor, and that's with respect to a particular target. So we would like to maybe use this data for things like labeling stuff or coloring, but how again can we associate the two? That's really the goal here. So I'm going to show, well, just really fast, some code where I make sure that I only look at fields that have an action type defined. If you've ever dealt with messy data or things that are not defined, you might have like null values, but it's really fast and easy here. I just get rid of those. I select for those action types. I do that unnest trick, basically, that I just covered. I aggregate it, so I'm only taking the first action type. There could be more than one. For this demo, we just need something to label with, but this is very quick and easy and painless. And then I finally join it to the molecule table. And we're going to use that later, and you're going to see why it's useful. But also just to touch upon this, we started with 12,000 plus change molecules, but then after all the filters and joins, very quickly we're down to about 4,126 pretty good quality ones that have exactly what I need for this demo. So that's pretty nice. So these smile strings, I think I briefly mentioned them. I also have a link somewhere, but how they work is that there are these strings that you can use to represent molecules and the chemical compositions. There are tools available for you to do things like draw the molecules from those strings and also take similarities based on those chemical compositions. So it's very easy actually to pull it out in this case. I'm just selecting for it and then I'm execute. This is when I talked about lazy evaluation before. This is when I'm like, okay, time to stop being lazy. Let's go ahead and grab this and put it into a data frame to start working with. So this is how you can kind of design your queries and then finally execute them. Now, first of all, I just wanted to show what you can do with RDKit. I just took those strings, just grabbed some of the molecules, and you can just very quickly just plot them. You can visualize them. You can use it for all sorts of things. But we can also create these molecular fingerprints, also using RDKit. And this is what's going to be used to derive similarity. I'm not sure how many of you have seen like a similarity matrix before, but here's one, for instance. And what this is essentially telling us is like how similar two of these are based off of this chemical makeup. But this is an interactive plot, which I really like. So you can kind of zoom in here and okay, well, it's kind of hard in this device. Sorry about that. And oh boy, I'm fat fingering this, sorry. But you can go around exploring it. So this is a very intuitive, interactive way of just taking that data and just tossing it in there. So imagining an interface like that can easily just give you these things. Secondly, I tried clustering. I'm like, well, now that I have a similarity matrix, can I start to see how they begin to cluster together based on these drugs? So if I can hopefully get it right this time, come on. Well, here's an example, right? Okay, so I don't have a background in pharmaceuticals and I have a hard time pronouncing these, but you can see right here, like intuitively, two of them, I'm pretty sure they're related, at least they share the same name, and based on their chemical compositions, it does think it's the same or similar. We can see it, if you've ever seen a dendrogram before, this is showing it's like sharing like a common ancestor. So this is a really quick way to also cluster and visualize like your drugs or any other kind of thing you want. So there's a lot of advantages, like I'm saying, in terms of combining data with visualizations. And now there's UMAP, which I won't cover too much just yet. We'll have the other notebooks to talk about it. But it's an algorithm to basically allow you to take a bunch of features, for instance, and embed them into a lower dimensional latent space. This is great when you have a bunch of stuff, but you want to like kind of visualize proximity or when things are similar, right? Like that space before. How do we take these essentially, like if it's like a 4,000 element row, that's like a 4,000 dimensional thing. And it doesn't really live in that space. It's lower dimensional. So this allows us to compress it down to 2 or 3 dimensions to easily visualize and also use as features. So I'm just quickly making an embedding from the similarity matrix itself. It's actually really efficient. So I was able to take a bunch of, like all of them. Before you were just looking at subsets of the matrix because these algorithms are kind of slow, but this one it's like zoom. So let's go ahead and actually take a look at this. And this is also where the labels are coming in handy. Sorry. Because what I ended up doing is, because I joined on those tables, we have an extra layer of visualization here. We can see that they can kind of cluster according to action. And we can also move around. I attached a lot of extra data from that table. So if you mouse over them, and you're all free to do this, by the way, and oh, geez, that pointer is really giant, so you can't quite, and I can't quite see it. So it's gonna be, I'm on hard mode, I guess, today for presentation. Sorry about that. But I swear there's useful information in that thing. And just imagine how useful that could be for a researcher or something to be able to just pull this up real quick and just try to look at how they aggregate based on similarities. And I'll move on briefly, but I just think that it's really interesting in terms of how these cluster. It's not, I'm only using some basic embeddings, so we want to take this with a grain of salt because molecules that are close together in this space are not necessarily identical or similar. It's just based on that feature we used. As we explore more of these features, we'll see that it allows us to visualize similarity on different levels. And that can have a lot of advantages, like when I talk about gene expression in a little bit. And I think that's where I want to leave off for this notebook, just trying to give you a high-level overview. Any questions so far, or would you like me to move on to the next one?
Any questions? Or any thoughts? Anything that seems like, it's something useful, not useful, would never do, would do? I don't know. Give Jordan applause maybe. Oh yeah, go ahead. Great. Yeah.
Well, hold on a minute.
It's kind of new to Ibris, and just want to understand the data you downloaded. Do you download that locally, or how
Yeah. So in this demo, I was just playing around with it in my notebook. So I did download the Parquet files directly to my laptop, but you have many ways in which you can access the data. So it supports, for instance, remote access. So it doesn't have to be on your laptop. And that's probably a good idea for some very large databases. You're not going to be able to store it all there. And you can also access databases directly this way as well. So there's many ways you can use IBIS.
how's it all secured?
So I can't comment on that. When Fernanda comes, I think she can give a better explanation, but I'm pretty sure yes. Just because the back end is essentially what's handling all of that, right? So if you go through something like, oh geez, I can't remember if Hadoop is an example of it, but you have different backends you can choose. So if you shop around for the one that's most secure, then you can pair that with IBIS so that it runs on that.
Thank you.
Mm-hmm. Yep. Okay, so the question was, do you have a sense of a difference between IBIS and polars? Well, the good news is you can have both. You can intro convert. I would say what's nice about polars is, again, it's I have used it a little bit. I like it a lot better than pandas, I'll tell you that. But they go well together. The languages are very similar. Polars, if I wanted to execute and turn it into a data frame, I'd probably want to manipulate it through polars at that point. IBIS works through basically the queries, right? So behind the scenes, like DuckDB, it's making SQL queries, and that's great, but it's still not working on a data frame. It's really working on the database, but it gets most of it done that way. But when it comes to execute, that's when I would suggest polars or something if you want to use it for that. Does that help answer? Okay, well, let's move on. Here's the next one. So now we're going to explore disease embeddings. And the goal here is to grab it, again, from that database. Again, it's locally here, but you can also access it remotely. And also you can use queries to do it. And I'm just loading some functions that we're going to use later. So I'm not going to go too much into detail about these things, but essentially I just wanted to make sure I had some great ways of visualizing it. So I did a lot of the legwork early on. So let's just go ahead and go down to the data prep. I'm just showing you the diseases table here. And again, you're free to go ahead and browse around, but I'm going to go ahead and use this for an interesting example. I'm going to actually start talking about targets first, and it might sound weird because we're talking about diseases, but if the goal is to start associating the two, why don't we have ways of representing diseases that way? Because if two diseases have similar targets, or shouldn't they be similar as well, like diseases? In terms of gene expression, I mean. So the goal here is, can we visualize that somehow? Can we aggregate diseases based on what known evidence we have for associated genes and the ways that they're expressed? So that's the goal here, but it's a pain normally because this is all deeply nested data. So I'm just showing you again an example of the table. That's very deeply nested. So I'm just using those tricks again to unnest it, just like this. And what I want to point out is, let's see here, do I have an example of it? One moment. Okay, anatomical systems. So these targets, they have multiple labels, so you can aggregate across different ways. For instance, we can have it based off of where is it expressed in the organ or where is it expressed in the overall anatomical system. These are great ways of labeling and also aggregating. And that's exactly what I'm going to do here is I'm going to essentially aggregate the gene expression of the targets based off of what system were they expressed in. This is very high level, which is why I chose it, because if you have like 1,000 labels, good luck trying to visualize that first of all. But I think that it's just a good thing to try out. So what I ended up doing was I vectorized this. Again, using IBIS, I just took for each target, where were you expressed? I unnested all that stuff, turned it into a table. I turned it into, I did a pivot, if you're familiar with that, and so that it's now like in a common format. And you can kind of see it down here now. I'm using Z-score, but you can also get like, what is it, reads per million or something normalized. So you've got a lot of different values you could aggregate and normalize on. But here I'm just showing that for each now, for each target. This is an ensemble ID, by the way. So this is associated with a protein coding gene, or transcript rather. For each one we have now, we've aggregated over the expression values, their Z-scores, and there's quite a few of them. So this can be used now as a vector to embed, for instance. Remember how I said high dimensional data? And it's also something that we can now pair with the diseases. That's because, like I was saying, there can be several targets associated with your disease. So again, treating this like tables, you can join on that and say, for my disease, and for the targets associated with it, I would like to represent it now as also an expression vector over in anatomical systems. In this case, I just take an average, right? And this is going to come in handy later. I promise it will make some sense. Finally, I'm just also grabbing some labels out of this, just so we will have a nice visualization later. I'm just asking, okay, so once you average over all of it for your disease, where are you most highly expressed? For instance, what system? And this will help us visualize later on with coloring, but it's also a potential label you can predict on. And here I'm just showing the end results. So now for each disease, I'm like, okay, I don't know which one that particular is. I don't memorize these codes, but it's like you're associated with connective tissue in the sense that of your targets, you are most highly expressed in tissue. There, I'm just pulling together the final query. And now I'm actually going to start doing these embeddings. And you have a lot of choices when it comes to using UMAP to doing embeddings. I won't go too much into the nitty-gritty details, but in this example, these diseases have descriptions, raw, free text, and you can use that to do embeddings. I'm using scikit-learn here. If anyone's familiar with it, it's a wonderful tool to use. It's in Python. And I'm using that because I want to vectorize the text. I use term frequency, inverse document frequency here, and it really handles a lot of it behind the scenes. And I also normalize it. So this is essentially what this is going to do is it's going to chunk up that text, vectorize it, and then it's going to pass it over to UMAP, which will embed it, and then we'll be able to visualize it. So that's great, and we'll be able to do that with any text, by the way. Like I'm showing it with this, but you'll see too, we'll do it with genes and other things. The other thing is I wanted to also take those expression vectors I just talked about and embed them too. They're already vectorized, so we can just toss them right in. So that's what I'm doing. So now let's actually talk about what these visualizations are. The first one is the disease descriptions one. Again, I've paired those labels that I was discussing before. So this is for each disease. Okay, like, let's see if I can pronounce any of these. Okay, that's not much of a disease there. So that's another thing is that I didn't necessarily filter on other terms. It could be that to get better quality results, I should study the data more and think about other fields I can filter on. I just took everything and said, what's it going to look like, right? So like, okay, pseudobulbar palsy, for instance, it's most expressed in the nervous system. I don't know if that's true or not, right? So we are just, we're taking some aggregates, we're taking a max, we're finding where it's most expressed. It might be true, it might not be. I don't know much about these things. I'm not an expert in this area, but you can very quickly visualize these things. And this is just on the raw text alone, which is kind of interesting. And you might see some clustering behavior. I might zoom in a little bit. Oops, or if I get the hang of this here. But where I want to show you next is where it really kind of pops. And this is using those embeddings I was talking about. Now you can really see, oh, okay, I was even embedding this before, I mean the expression ones. You can really see now that there's this clustering behavior going on, right? And this is based off of gene expression. So it's, first of all, really exciting to take a look at it. You can look at the neighborhoods, you can see maybe okay, I'm a really bad driver here, sorry about that. Because one thing you might notice sometimes is that very similar diseases will be next to each other. I didn't really look ahead of time to see some good examples, but you all have access to this. You can all explore it. Just note it's only embedding on gene expression. So as far as the algorithm is concerned, you're similar if and only if you've been expressed in similar, if you have share similar gene expression profiles based on the targets you're associated with. But it's a great way, again, to visualize data on the fly, and these are going to be used as features later on. And in fact, that's one of the things I'm doing here is I'm exporting these mappers because I'm actually going to use them in that final notebook. Any questions before I move on to the next one? All right. Oh, yes, good question. So let me go back down. All right. What? Oh, I was just trying to, oops.
Go back to it.
Yeah, okay, so, wow, this is really sensitive, sorry about that. Okay, so the question was, what are the axes? So how UMAP works is you can choose to embed in 2 or more dimensions. And the dimensions themselves don't necessarily make sense in the sense that it's like a particular variable. It's that it's a combination of these variables, right? So in this case,
That's the machine learning.
Yes,
bit.
exactly. Yeah. So in this case, I told UMAP embed into 3 dimensions, and then I was able to plot it that way. You can also embed into 2 dimensions, and for like very deeply or for data like this, it probably wouldn't hurt just to go down to 2-dimensional because I'm not seeing much like separate clumps, right? So you might get away with it, but you can go higher too. So I guess that's just a long way of saying that the axes themselves don't necessarily carry intuition other than the fact that it's supposed to express a latent space in which these are all embedded, where if you're close to each other, it's because you have similar anatomical system expression profiles. Any other questions? All right, moving on.
Keep your energy up. We're still going to do data ethics after this.
All right, so we're going to give a similar treatment, but this time to the targets, and specifically, I am looking at the protein coding ones. And first of all, this is just a prelude. It's very similar to the last one. I just have my little helper functions here. I'm doing some data prep, and the first thing is I'm going to take a look at these subcellular locations. This is based off of where it's being expressed in the cell. So these are also associated with gene ontology terms. I've provided a link out to a nice site that explains it if you want to learn more. These genes can be associated with 1 or more subcellular locations. In the case of this demo, we're just going to look at the first location because I want to use them as labels again. So just know this isn't perfectly representative, but it just gives us a way of just quickly visualizing it. And I just, again, want to highlight how easy it is to do that because once I aggregate and group by my IDs, I'm just like, give me the first location. Right, so I really love like grammar set up like this because it's like you're talking to your data and saying, give it to me like this. So it made it very easy for me to now create a table in which for each of these protein coding transcripts, you have the first associated location, right? So this one is most expressed in the mitochondrion, for instance, whichever that one is. And I'm going to use this for later. I'm also going to use those Go term descriptions. So if you haven't heard of what Go terms are, these are gene ontology terms. They help describe, for instance, like the functions of your gene or other kinds of aspects. They're pretty useful for all sorts of things, but we can also use them for embeddings. We can use them as raw text. We can use them as like a binary indicator. In this case, I'm just going with the raw text version, which is the fact that for each of these Go terms, well, there's a description associated with them. Also available on open targets, which is convenient. I'm concatenating them. I'm just joining them together. So if you got 10 Go terms associated with your gene of interest, you can treat it as a document by just joining them together and then using that tokenizing trick that I talked about before. You have other options too, like I was saying, but this is what I was doing here, just to explain on a high level. And I'm going to combine them into a final table so I can start working my magic. And if you want, you can scroll around at some point and take a look at it. And then I do query execute to put it into a data frame. And then the last prep I'm going to do is I'm going to use another library. It's called PyEnsemble. It was really great for me to find this. Sometimes I struggle sometimes to pull the data I need from like other databases like NCBI or Ensemble. So this is a very quick way of doing it. And it just allows you to, for an ID, like the one up here, grab the associated transcript. And that means you get access to the nucleotides. And using BioPython, you can also translate into a protein. So there's already a lot you can do just by pulling this data in. This is why I love how highly connected it is. And I just have some descriptions here. If you want to go back and look at it, that's fine. Just know that this is what I'm doing as I'm pulling it in, getting the nucleotides, and also trying to translate the transcripts. It wasn't perfect because I was just doing a quick and dirty demo here, but I am demonstrating how feasible it is. All right, now going down to the visualizations. We're going to do the same thing we did before. So those gene descriptions, same idea. I use term frequency and inverse document frequency, turn into an embedding. Gene descriptions, same thing. Each gene has a description, so you can just vectorize it. And now I'm actually, okay, actually we started with gene descriptions. Sorry, I thought we were starting with Go term descriptions. But yeah, so gene descriptions, they're not that long. Right, so it wasn't very high quality. We can already, oh wow, I'm going off into the stratosphere here, sorry. Oh, okay, wow, got a mind of its own. We can see some clustering behavior here. And again, this location, this comes from that subcellular location, the first one it appears in. So again, it's very handy for visualization. We can see at least for nucleus, which is okay, yeah, not a big surprise here. They're mostly clumping together in a way in which kind of makes sense. And this is just based off of text again, just based off of how it was being talked about, right? But we see a little more chaotic behavior elsewhere. So it's not necessarily going to be perfectly reflective of an embedding that describes if you're expressed in the same subcellular location, you should be close to each other, right? This is off of a different method, but it also helps us compare and contrast. Oh, and by the way, like these, you can go ahead and filter if you want. Like you can double click even to isolate. If I can click fast enough. Well, there you go. Like, so if you want to explore, you got a lot of options here. And for those that aren't familiar, this is Plotly. I love Plotly, if you can't tell. I've been using it a lot. There's also Dash, which is something that Plotly supports. It allows you to, spin up your own web servers and do a lot more, actually. Behind the scenes, it's using JavaScript and all, but you don't have to worry about it. You can code mostly in Python, take in this data, make really awesome visualizations and interactive things that researchers can use. So I think it's practical, and I just also wanted to just plug for that because it's just great to find these kinds of tools available, and it's also open source. Now, here are the gene Go terms and their embeddings. Same idea, I'm just vectorizing it. These are a little more, if I don't go too fast. Well, we're seeing a different kind of, oh no, come back. So part of it is that when you have a lot of data and if there are limitations on like your GPUs or something, it might like try to catch up, right? When you zoom in, so you're waiting and go, it's slingshots, right? So it might be a little difficult for me in this demo to show you exact things. I apologize, but same deal. We're just visualizing them and we're going to use them for later. And now we're going to some interesting ones. So those nucleotides, I retrieve transcripts, right? And ATCG, for instance, and we're going to use chimerization here. So we're going to break it up into chimers. How it works is you can think of it like a tiling over your nucleotide sequence. The first one is just 1 grams, which is just ATCG, right? So we just break them up and we represent your sequence as essentially like a vector, a count of ATCGs. That's it. But we can also do 2 grams, which is like if you go over your sequence, and if you have a sequence ATCG, then it does AT, and then it does CG, etc. So it gives you ways of also describing long range behavior, and we also treat that as a vector of counts. And you can just keep going, but as you get higher, it becomes more combinatorially explosive. So I'm just limiting it to a range of 1 to 3. Again, sklearn's TF-IDF vectorizer takes care of it for you, which is really nice. So I'm just saying, take n grams from 1 to 3, like so it's going to take these plus the 3 grams. I've included a link in case that was like unfamiliar to you. I don't necessarily have the time right now to go into how it quite works, but just know too, this tool handles a lot of it, which is great. And I'm doing now these transcripts. I'm going to go ahead and visualize them. And you're going to see some striking differences here. This is more of a continuous space. First of all, because now we have access to a lot more like features here, because I didn't count, but these are sparse matrices of like sometimes like thousands of dimensions. In this case, I don't think it was that many, but we get a smoother kind of embedding. It's less reflective of the location, but we also see some interesting kinds of aggregation behavior. So like over here, you'll notice a lot of olfactory receptors, like the family itself is actually clustering together. So what this is telling me is there's probably something conserved in the nucleotide sequence, and that's why they're all clustering together, right? And so UMAP is trying to be strategic in how it's doing the embedding, and it tries to keep those really close families together, and that's what you're seeing here. There's a lot of other interesting structure. I'm kind of afraid to go in there because I'm probably going to slingshot, but feel free to explore. There's a lot you can do. And I'm just showing you also how this works with proteins. Same deal. It's a little more squished here, a little more conserved, but you also see the olfactory receptors, again, sticking out like a nose, I suppose. So these are just things that you can go ahead and explore. And let's see. And now I'm going to go ahead and export them. So we're going to get hopefully to some of the interesting parts now where we use these embeddings. But also just know that this is just great for debugging, because as I was going along to understand the data, I added things like my indices and other kinds of useful information that isn't just helpful for me, but a researcher would probably want to see something like that too. Any questions before I move on to the last part? Alrighty. So for this next one, I'm just training a dumb model. I'm just trying to see what we can do with it, right? So I want you to take this with a grain of salt. This is not necessarily how you should do it, but I'm trying to, again, demonstrate the ability of ingesting a large amount of data, iterating through it, and trying to train something using data from Open Targets itself. I think I already touched upon the association evidence, in the sense that for each gene and target, we can have evidence from multiple sources and score that association accordingly. This is great for discovering potential drug targets, because if there's a high association, maybe that's where you should look next. So what we're going to do is we're going to train a model to try and predict on that. We're going to ingest all those embeddings and then all the data, a lot of data from the tables, from the association tables, and train on those scores. We're going to predict on them. The idea is not to get it perfect, though, because we're trying to see how it will evaluate things it's never seen before yet based on these embeddings. We want to score things that we maybe don't have much data on for gene associations, and that's what this is going to facilitate. Again, I have a prelude. I'm not going to get into it. I had to have a couple of helper functions. I had to load up all those stored models. I mean, ideally, you don't want to pickle things all the time. It would be nice if you have them somewhere else. It takes some time. These things can be large too. But just for this demo, I saved everything from all the other demos and just loaded them this way. Same thing with the tables that we need. I'm just giving you an example of these tables, because what I'm going to do is, okay, yes, overall direct association scores. So Open Target stores association scores in different ways. This is this direct, like they've got a special sauce sum where they just take all the scores and sum it together for each of these associations. But if you're more interested in like, well, what are the associations with just publications or through genomic pathways or things like that, you can break it down on that level too. There are different, there's a different table for that. So using something like this, you can filter on that. But for this, I'm just looking for a score to try and predict on, right? So I'm just taking this, joining it together in a way that I can actually label it like train test and shuffle it so I can actually use it for my, my very simple machine learning model. I'm going to then extract all those embeddings. I was very careful to make sure that the indices lined up so that for each of my, for instance, genes or targets, it's the same index for each embedding. So I just clump them together. I transform them with softmax. You have different ways in which you can transform your features. I won't necessarily get into this, but it approximates a distribution in this case. We're going to pretend it's like kind of like a joint distribution, and we're going to predict on like, hey, given this disease, what's the likelihood of these genes being associated with it? And that's kind of like the power of things like this, is that you can kind of embed and train that way. And I'm just using a simple model here. It's a, geez, I can't remember it. It's a gradient boosted random forests. I think it's light GBM. I think it's supplied by Microsoft. I can't remember, but you've got many different models. And one of the important things about this is that once I've done all the hard data wrangling, these things can be fed into pretty much any model that supports these sorts of formats. So you're free to explore. So I'm just iterating over it chunk-wise. I was just pointing this part out. This is like overkill in this case, but if you're dealing with a lot of data, you don't necessarily want to transform all your features at once. So either you want to call it and have it be done remotely on the data there, or you want to like chunk it up and iterate. So this is just, feel free to come back if you want to look at what I was doing. I'm just finally getting it ready. So I have my train and my test. And then now I define my model. I'm really not bothering with tweaking it much. I'm using cross entropy here. I won't get into that, but it pairs well with the distribution. In practice, when you actually want to use this in like production, you should try to work on exploring your options, your hyperparameters, and tuning it to get a better model. And I won't go into details about that, but there's this thing called grid search, and you can, for instance, try and optimize on choices. Because, like, for instance, those embeddings I showed you before, I was choosing these different metrics, like Hellinger or Euclidean, and part of it was arbitrary. Part of it is because I kind of understood like the shape of the data and what might work well. But if you really wanted to make sure you were doing a good embedding, you could do like grid search, and you can try out different embeddings and score them and just pick the best one, right? So you've got a lot to explore. This is my model. I'm just showing, it gives you this nice little graph. This is a dead simple one. You can chain them together, do all sorts of fancy things, use a neural network if you'd like, so a lot of options here. All right, so what am I doing here? I finally did a train test real quick. I'm just doing some quick metrics. If anyone's familiar with like R squared, that's a metric that can tell you like closeness of fit really. It's not perfect here, right? But that's also not the goal. I don't want to exactly predict or overfit to this thing because I'm trying to, predict things maybe it's never seen before, right? Just get it so we don't want to fit too closely to the data. But in practice, again, you got to be careful with that too. But now for the visualization, what I'm doing is I'm just grabbing the index for Alzheimer's disease based on its gene ontology Mondo classification. And then I'm using that to now run my prediction. The idea is that once I've trained my model on what it's seen before, it's now going to be able to range over targets in a way that it makes predictions. It's like recreating the scoring function sort of, but only based on the embeddings, right? So it's limited by the features you give it, and it's saying like, well, okay, based on like gene expression you gave me, how can I score its relevance, right? So this also gives you more fine-tuned control in terms of how you want to describe correlation. So we can go down now to the heat map-ish. Like what I was doing is I was taking one of those embeddings from before. This is this is the gene descriptions. Remember that thing? So I was embedding via gene descriptions just like last time, but I've changed the color. And this is nice because the color now is the prediction score. So your eyes can immediately like pick out some of these things. And like, I'm not going to pronounce that, but I've looked up some of these. They're definitely associated with Alzheimer's. I'm not going to say go and run in and explore those because this is a dumb model that I was training, but this just really captures the power of what you can do. You can start exploring it and visualizing it. And this is also where it really helps now to like pair program with an expert. I'm like, is this making sense to you? I can show them now visually. Hey, isn't that something? Right? That's really cool. So there's a lot you can explore, and this is just one disease, right? I could call different ones. Unfortunately, I can't do it on this one. This is an HTML. Like I just exported it, but you're free to explore it in different ways. So it's just really powerful. There I go again up to the stratosphere. Oh boy. Another thing is that I can filter. So you've got a lot of control for these things because maybe I'm like, I don't want the low scoring things. I just want the good stuff, right? So again, using like IBIS, I can just go ahead and look at just the highest scoring ones and the top 1% here, right? And we can see some clustering behavior. Oh, those are olfactory receptors again. Hey, isn't that interesting? You can also see this is another hotspot. So all of these are actually pretty high quality or predicted to be highly associated with Alzheimer's disease. So that really helps filter it out and narrow down the possibilities for researchers. And there are other ways I can make this available. I can give you a list, sort it, but just like ranking, just like Google is like, hey, search for this and give me back the top results. So you don't have to just use visualization. I just find it incredibly powerful. And I think, and then this is, if you'd like to explore it, I showed a kind of, in the first case, what are the top results, kind of like a query, right, from my model in regards to the predicted score. One thing I want to point out here is that a lot of these are very similar, at least in regards to my features, right, like with gene expression or nucleotides. A lot of these are very, very similar, and so the model is predicting very similar scores. This can be a concern for a couple of reasons, because, like, for instance, if you want a representative data set, then it's like overrepresentative if you have too many of very similar things. So clustering is an option, and it's also something UMAP can do. So that's just something I want to point out here, is that you're not getting a lot of diversity of results here. You have to dig deeper. And so I just wanted to point out these are important things you have to deal with real data. And now here is the table for associated scores. This is being pulled directly from the ones from Open Targets. I'm just sorting on best score. I'm also pairing my predicted score from the model to it, so you can kind of compare and contrast. And it's interesting because like sometimes it will predict something very low for what would technically have high evidence. And this doesn't mean that it's wrong. It's limited obviously by my features that I chose here, but it gives us a different way of thinking about and comparing and contrasting data. And I think that's pretty interesting too. And so yeah, this just gives you some ideas, hopefully, of what you can use a lot of these tools together to do. And that pretty much concludes my demo. So if you have any questions, I'd be happy to answer them. Otherwise, back to Anna. How do I switch back? Thanks.
Thank you, Jordan. That was really, really nice. You guys can see I'm so lucky. I don't know, I was smiling the whole time. I'm so lucky to work with such a nice team. You guys have questions for Jordan? I think I saw a few hands up.
I'll also be
Yeah,
available to
oh, back there.
Yep.
Do you want, here, I'll give you the microphone. Oh, you got one? Because I think people in the internet.
Thank you. Nice talk. My question is about the dimensionality reduction.
Yep.
I'm not sure when you talked about the filtering, you meant it's filtering the dimensions or filtering the data itself. And about the dimensionality reduction itself, so you touched at some points about the clustering or different techniques that we have, but do you have any thoughts on that part as well? Would be great.
Sure.
Thank
Do you want me to explain like dimensional reduction a little bit more and then talk about like clustering and stuff? in
So
regards to
up
that?
to you. My question more is about what techniques do you use for high dimensional data that you have?
Right. So there are definitely a lot of ways in which you can do dimension reduction. Even just simple eigen decomposition allows you to do that. So I like UMAP in the sense that it's pretty speedy for what it does. I would say the embeddings are pretty decent quality. I have no way of necessarily comparing and contrasting in this example. I would need to compare the algorithms, but I would say that the way that it works, it's fairly useful. There are some things you have to be concerned about in the sense, like for instance, there is a bit of randomness to this. It's stochastic. You can fix the seed and to deal with it, but what this is essentially being used for is that in the example where I was, for instance, doing disease descriptions and taking those words, well, each word is being represented as its own dimension. That's kind of crazy. I mean, like, you know, if you have like a 10,000 dimensional space, it's not necessarily that you're going to expect your genes or diseases to kind of cluster in that space. So really one of the reasons I would want to use UMAP is to embed it into a lower dimensional space that captures that similarity as if you were working in that higher dimensional space. But the second advantage is compression. It's really nice to have lower dimensional like features that you can feed into models. It can save time, it can save space. It might even be higher quality. You might get better results. So there can be a lot of reasons you would want to explore it. I didn't get too much into clustering. It wasn't, in this case, it wasn't necessarily clustering. You were actually seeing just the natural, like based on the similarity metric I was using, it was just naturally aggregating. And the colors you were seeing was my choice of labels. Now there are things you can do like KNN, like nearest neighbors, things like that. You can cluster. UMAP supports it. Of course, once you get into this format, you can use your clustering algorithm of choice. So you're not really tethered to UMAP. So there's a lot you can explore. But I would use clustering, for instance, when I might have a lot of like a hierarchical thing, a lot of representatives of like subunits, right? So it's like, I don't want to represent them separately in that case. So clustering might help with that, like to quote, unquote, deduplicate, right? So there's a lot of uses for clustering. Did that answer your question? I'm happy to talk more to
thank
you. Okay. Other hands? Yep.
The ensemble IDs for let's say Swissbot IDs, because there isn't it's not exactly one-to-one in fact. So are you Are you doing that parsing from Ensemble and the Go terms from inside Ensemble or inside Open Targets?
So as far as the Go terms are, they have a table available to you in Open Targets, which is convenient, and it's matching the ones that you can access
Sure.
elsewhere.
Okay.
But the Ensemble ones, yeah. So I was just, because that field was available, I was
Yeah,
like, let me
sure.
just pull it. But I can't quite remember what the other ideas there, but if it were available, you can pull it that way. But otherwise, you would have to do the dirty work of trying to look it up and find it. So it can
Okay.
get involved. I guess this is low-hanging fruit in this case.
Anything else? All right. Okay. All right. So you guys are free to explore and then come chat to Jordan or myself or the rest of the team. I think we have a few more slides to finish up. I think we're not quite finished. Oh, the clicker. You want me to use the clicker?
Good timing.
So now that we've sort of done something, I think we actually have a few slides to finish up the demo as well. 1 second. Let's see. We did this. We did this. Okay. Jordan, do you want to wrap up some of these topics?
Yeah, so quick takeaways from what we were doing. There are tools today that can make analyzing large data sets easier. And like I was talking about before, you don't have to struggle so much about in-memory processing when you use things like lazy execution. And I just demonstrated a couple of those tools. They're part of a greater ecosystem, the Arrow ecosystem. Think Arrow in, Arrow out. Is it their slogan? But anyway, it's something to maybe start looking into. If you even start learning these tools, you'll probably be in a good shape if there is going to be a point at which you want to do like distributive like processing. Storing files in a columnar format, Parquet in particular, I just wanted to like advocate for how useful it is. I really do see that as an upcoming trend. As a computer scientist, I was super pleased to see something like that available. It's really hard to find really good data like that. Avoid CSVs if you can, and proprietary formats are really difficult to deal with. So if you can find ways of making it open source, that's great. And I would say visualization is essential for understanding your data and also what your machine models are doing. Because I was showing you that prediction, I could start seeing, and when I was starting out, I was messing up, and it was clear to me. I was like, well, this isn't making sensible predictions. So you can really use it to help drive your development on top of just making it available for researchers to use. And I would say that it's really necessary to combine computational and domain expertise, right? So I didn't know the ropes here. I was doing my best with open targets, but it would be even better if I paired off with someone. I would say if you ever want to look into it, consider replicating the infrastructure and the design of open targets, because they have it freely available. You can download it, you can do your own web server, and it's just a great way of I think, making this available. And actually just consider also contributing and supporting efforts like this and to open data in general. I think that's really what gets you to the point where you can make really important scientific discoveries, and it gets you attention, right? I wasn't trying to like plug in open targets, but I was like, finally a good data set. So people are noticing, right? People are going to notice when you make good quality data available. It really helps.
All right. So there's actually one more thing that I wanted to cover, and that is on AI ethics. And so I actually got into the concept of AI ethics, I guess, when I started working at Bioteam. And one thing that I wanted to convey is that I myself, with my very deep interest in the topic, have gotten so exhausted from all the things that are coming out that are so useless, in my opinion. I am a person of utility and forward momentum. And to me, I guess this is just to validate if any of you are sort of struggling with the same concept in your companies or teams, that these concepts of ethics are becoming sort of performative in a way, right? People are starting to talk about it just to sort of show or peacock, let's say, the concept, versus actually making any progress or actually implementing anything tangible. And so I guess I followed way too many people on LinkedIn or something, but I feel like every day my whole posts on LinkedIn are now about like AI ethics frameworks and like articles. And now I think ChatGPT must be generating all these articles or something. And essentially the point is the field is now very crowded without any actual actionability. So if you feel that way, I guess I would just like to normalize that as a feeling. And then all to say that we still have to, as a community, find some way to make progress, especially because as we use these higher order models and as we sort of start using models that are essentially taking a lot of unstructured data and giving us feedback, we actually do really need to have safeguards in place for our teams and for ourselves, and of course for our companies, to actually say how we thought about designing and structuring our data and models to be in some way ethical or equitable or to mitigate bias. Because if you look at the publications, we did a huge assessment one time of scientists and their understanding of bias in AI/ML, and essentially what I would say people said is that, listen, like, data is biased, people are biased, AI just sort of mirrors us, and so, you know, yeah, things are going to be biased, and there was this general sort of override of bias. What I have to say is that when people actually assess these models, the models, because they're taking some general consensus or statistical average, they actually really amplify bias a lot. And it's actually, even for a person who's sort of like, I don't like to dwell on things, but I do have to say that publication-wise, bias in these models is an actual problem. And when we think about making medical decisions or designing drugs, it is actually going to cost us long-term progress. And so there are things, and I also actually included, I included 4 topics. So essentially accountability. Again, that AI ethics at the moment is really very performative. And most AI concepts at all are just not really even understood in order for teams to feel accountable about them and for leadership to really say whether or not you have accountability in your teams, right? It's really about the people who are working on the data. So as Jordan said, I think that's one of my favorite qualities about Jordan is that he's never going to overstep his capabilities. And so he would always choose to say less and be really accountable for it than to say more and not be able to be accountable for it, right? And I think it's those kinds of people on your team who have to be responsible for making sure that you're documenting your data, your models, the way that the models are trained, written, and things like that. And you know, at the moment, bans on things like ChatGPT are very trendy. I have personal views on that. As teams and companies, you have to obviously decide what your sort of internal views are on things like that. In my opinion, it's obviously not a long-term solution. It's not really going to get us anywhere to try to ban some of these technologies, but to really add in some layer of documentation around what we're doing as teams. If I'm the scientist and Jordan is the data scientist, we have to somehow document how we trained this model, why we came up with these parameters, how we tuned it, some sort of model card. I'm actually already sort of exhausted by the concept of suggesting ideas because I think it has to be really unique to your team. Like, there's only some things that your team is going to want to do and actually do that another team is not going to want to do and actually do. And it really is an internal discussion. But there are lawsuits underway, which I think will provide some sort of like legal framework for us. But I do have to say, legal frameworks are not always suited to scientific workflows, right? Or really to anything. I'm going through a divorce and I think legal frameworks are ridiculous. So anyway, but you do have to think about your value and alignment situation as well. And also really actually do think about fairness. And I think that even just documenting in your processes that you had these conversations as teams, as research groups, you know, even just the documentation of the discussion of it is going to get you so much further than people who sort of just, I don't know, just put it on their website or whatever it is. And there is research sort of going on at the moment of how to help models unlearn some of their bias. And so teams are looking into it. So keep on top of the publications. I guess try to filter away sort of some of the discussions, you know, like UN, World Health Organizations, they all have these sort of concepts of do no harm, people first. They all become iterative. Really look at the research where people are actually scoring things. We're actually putting some frameworks into place. Same thing with fair data, right? Like we actually need frameworks and scores and ways to measure this. And I know that from Bioteam, if a lot of you have sort of followed the Bioteam history, Dag used to always talk about and still always talks about putting onus on scientists to own their data, that it's not the IT's job, it's not enterprise IT, it's not the data scientist's job to own your scientific data. In the same way, I think ethics has to become this like really weird amorphous conversation that teams are having inside their research teams, and it's being iterative, that somebody just brings it up, documents it in some way, that when your team gets to something that could be production grade, for example, that there is a conversation that was had. And of course, a lot on data privacy and selection. We heard, again, some interesting talks from AWS where they're trying to create clean rooms. I think that's a really interesting concept and a really necessary one. So places within AWS where you can actually collaborate, you can de-identify information through, unfortunately, third-party vendors, which is its own discussion. Anyway, there are things that are coming into play where you can allow even your machine learning models to start visiting data instead of actually having access to data. I think that this is a really necessary development in the field. I just don't exactly know how it's going to play out, especially with the fact that they are proprietary or like individual companies that are de-identifying this information. I think that trusting things like that we have gotten burned, right, as a society to trust a company to protect information. Anyway, but that is really important. And also, I know that there's a lot of discussion around the concept of data ownership and stewardship and who actually owns data across large organizations and teams, especially with collaborations. I think some of those conversations, I guess none of them feel very conclusive in terms of that there's a framework of how to decide who owns data and who stewards the data. But those conversations really are important. And I have seen success of teams making those distinctions. Who's the product manager, data product manager at the NIH, for example. They have really large repositories, if you guys have ever visited, and they have to actually make those repositories interoperable, right? And when they make repositories interoperable, there's a huge discussion of, if I'm inside Biodata Catalyst and I own this data technically as part of this institute, and somebody's at a different institute and they own their data inside of Kids First or Anvil, how do we actually interoperate that data? Who now owns that data? How do we regulate the ownership? And I imagine those conversations are also really relevant to your teams, especially if you have teams in different countries with GDPR rules, et cetera, et cetera. I think that that's an ongoing conversation on data stewardship. And also on transparency. And again, there are people who are trying to add in some degree of explainability and transparency to these models. It's just not as simplistic, would you say, Jordan, as it sounds, right?
Yes.
And so I think the effort still needs to continue, even if the result is not quote unquote true transparency. And I'm not even sure that true transparency is the goal, and we're at 0 minutes. Anyway, the concept being, don't be concerned that if you feel like these discussions are sort of very nebulous, but you have to, inside your teams, still have them and give them structure. Don't be afraid to develop your own frameworks. There's a lot of frameworks. I included one here. We will have the slides linked and a good article as well. Anyway, thank you very much, everybody. I appreciate your 2 hours of time. We made it.